the hyper eating behavior that occurs with cannabis use is of great interest to researchers because there are times when you would want to stimulate appetite, for example, in patients who are treated for cancer. And here the researchers randomly assigned some lab mice, so that's our individuals, to 4 conditions at random. And so what does that mean is that we have 4 independent random samples, and they measure the feeding response in grams, so that's our quantitative variable. The conditions were either you give them nothing, the mice, or you give them cannabis, or you give them just the opioid blocker naloxone, or you give them a combination of cannabis and naloxone. And we'd like to know if there is evidence that the treatments, those scans here, significantly affect feeding response in mice, so whether there are differences on average in the population of mice. Because we have 4 independent random samples, here we're going to write our null hypothesis as the means for those 4 populations, hypothetical populations, because here this is an experiment. So mu1 equals mu2 equals mu3 equals mu4, and that the alternative hypothesis is non-directional here, just very generic. The null hypothesis is not true. At least 1 mu differs from at least one other one. So if we select the raw data and copy and then go to edit paste spreadsheet, if you do that, already done that, you get the data. So now how do you get the p-value? You go to ANOVA, one way. In this case, we have 1 column for each group, so it's by columns. Sometimes you have 1 column that gives you your response variable and the other column that tells you which group it is, in which case you would do it as groups. We calculate, and we get the whole ANOVA output, which again contains lots of information. For us, we're going to focus on the test p-value, which is 0.0197, so it's about 2%. We can also plot the data. So for example, in box plots, calculate, and we see immediately that the variability is very similar. We don't have a whole lot of data points, so we see the median is kind of all over the place here. And we can get the summary statistics with descriptive statistics, which is going to be important for actually getting the values of the standard deviations. And as you can see, the standard deviations are all very similar. The largest one at 61 is definitely no more than twice the smallest one at 47. All right. So we got a p-value of 0.02, so that's 2%. Let's check the conditions for inference. So we have 3 conditions. First one, do we have independent random samples? Yes, they're part of a randomized experiment. Second, do we have normal data or large enough combined sample size? All right, so the data do look like just variations of the same thing. They look kind of normal-ish. It's hard to tell whether that's a skew or just the randomness of this particular random sample. We don't have evidence of extreme skew. We have a total sample size of 22 with either equal or near equal sample sizes. So that's probably okay. We're maybe pushing our luck a little bit, but that's probably fine. And then we checked that the standard deviations were similar enough. So ANOVA is most likely okay here. So concluding context. Well, what do we do with the p-value like that? We're saying if the null hypothesis was true, results at least as extreme as those of this study could occur in 2 in 100, so in about 1 in 50 similar studies. So 1 in 50, that is definitely a bit surprising if the null hypothesis was true, but not unbelievably unlikely. So this is what we might consider some reasonable evidence against the null. The reason that when you write your conclusion in context, you want to cite that p-value is because words carry different meaning to different people, just the definition of a word. What is reasonable that doesn't mean the same thing to different people. Also, different people might have different standards, view different values as providing different levels of strength. So if you cite your p-value and you use a word that is not crazy for it. So for example, a p-value of 2%, you shouldn't say provides extremely strong evidence against your model, because that's not right. But if you say provides reasonable evidence, good evidence, somewhat reasonable evidence, fairly strong evidence, all of those are okay words to describe a p-value like 2%. All right. And again, if you cite your p-value, then at least you let other people be the judge for themselves. So here there is a reasonable, pretty good evidence that on average, the feeding behavior of lab mice depends on the kind of drug that they just took before, or drug combination, for the population of lab mice. Right? Can we say that the same thing would be true for humans? No, because humans are not mice. So number one, our physiology is different. Number two, we don't live in cages, which means that there are many things we do in our lives that also affect our responses, right? In fact, this is something important in the majority, by far, of medical research that is done on animals first and is promising on animals does not translate into treatments that will work in humans. So if you cannot make a conclusion for humans, then what's the point of testing in mice? Well, that's because you do not want to jump onto humans first. The first thing you want to do is you want to use an animal model to see whether that thing is horrible. So you're going to give a new medication. You want to see if it's going to kill your test subjects, because if it does, that's it, you stop. You also might want to test a variety of combinations of drugs or different dosage, etc. And so that gives you some idea of what might be promising.